perinatal environment. So many of the confounds that may be present in case control type designs can be overcome using twins. And twins also allow us to start exploring something else that's interesting about epigenetic variation, and that's the extent to which epigenetic variation itself is under genetic control. So we've just recently published, again this is work led by Eilish Hannon, a big study of DNA methylation across the genome in about 400 pairs of MZ twins and 400 pairs of same-sex DZ twins. And using standard quantitative genetic approaches, you can assess the extent to which variation in DNA methylation at each of those sites is influenced by genetic effects or by or non-genetic effects. And what you'll see by the kind of scatter plot there is that MZ twins are nearly always more similar. Each dot is a specific site in the genome. MZ twins are always more correlated for the vast majority of sites than DZ twins, highlighting that there's likely to be a strong genetic influence on DNA methylation patterns. So here's just an example of a really strong genetic effect on DNA methylation. Each dot on that scatter plot is an individual twin pair, and you can on x-axis is twin 1, y-axis twin 2, because there's a really strong correlation in the MZ twin pairs, a much more scattered plot in the DZ twin pairs. And then we can estimate the extent to which that variance is driven by additive genetic effects. And what we found is that these are quite widespread, and these additive genetic effects can be mapped to specific genetic sites in the genome. And so we're now identifying what we call DNA methylation quantitative trait loci, MQTLs. These are SNPs that themselves are associated with DNA methylation at a specific site. And you can see an example in the bottom there where genotype is associated with DNA methylation in a very additive way. Perhaps more interesting are those sites where it's not genetic, where there's a lot of variation, but that variation isn't driven by genetics. This is an example of an environmentally sensitive site, a site where maybe the variance isn't anything to do with genetics, but maybe driven by other things in the environment. So these catalogue of MQTLs allow us to start to explore the links between genetic variation and disease. So it's been highlighted from GWAS that most of the variants that are identified don't actually directly influence protein structure. So it's hypothesized that what they're actually doing is influencing gene regulation. And the other thing from GWAS is obviously these big regions of LD that encompass the GWAS regions maybe harbour many genes, and it's very hard to work out which is the actual causal genes in any of the genomic regions associated with disease. And so the kind of last phase of my talk is going to be on how we can start putting the genetic information that we're learning from GWAS studies together with this regulatory genomic data to start generating hypotheses about the function of those genetic variants. And I think this is a really important next stage for GWAS. So one thing that's clear is that genetic variants associated with disease are preferentially located in regions of the genome that harbour regulatory domains important for the cell type involved in that disease. So this is a paper that Eilish has in Press that's used partitioned heritability to look to the extent to which the genetic variation associated with a range of brain diseases is enriched in regions of the genome harbouring brain-specific enhancer elements. And you'll see that across the board, brain disorder, the genetic variants associated with brain disorders are enriched in these regions of enhancer activity in the cortex. And this is specific to brain disorders. So actually, if you look at non-brain disorders, those are the bars on the right, you don't see any such enrichment. So immediately, this tells us that actually it's really important to start understanding those regions of the genome that are transcriptionally active in the relevant cell or tissue type. So, as I said, we can start mapping DNA methylation quantitative trait loci. And so going back to our fetal brains, where we have genetic variation and epigenetic variation, we can essentially do a GWAS of DNA methylation and identify variants associated with DNA methylation. Most of these things happen in cis, So you'll see there's almost a diagonal line between position of the SNP and the DNA methylation site in the top left scatter point. And in fact, the DNA methylation sites associated with genotype often occur within 5 or 10 kb of the actual genetic variant. Perhaps more interesting are those where we see trans effects. So these are instances where genetic variation on one chromosome can influence markers of gene activity on a totally different chromosome. Again, this goes back to some of the issues with GWAS, because it tells us that the proximal gene to the SNPs that are identified in GWAS may not actually be the key gene that's involved in disease. So we must be really careful in interpreting those results. So these MQTL effects seem to be developmentally fairly stable. So on the bottom is a heat map just showing the effect sizes for MQTLs identified in the fetal brain, and then the corresponding effect sizes for different regions of the adult brain. So you can see very strong similarity across them. But I think the exceptions are always the really interesting things. And so here are some examples of genetic effects on DNA methylation that are specific to fetal cortex. So you'll see the first set of in red are fetal brain, where we see a very strong association between DNA methylation and genotype. We don't see any such association in any of the adult brain regions that we look at. And here's an example where we actually see opposite effects in fetal brain compared to adult cerebellum. So this is an example where actually a genetic effect on gene regulation can be both developmentally and tissue type specific. This is, I think, really a really important message for interpreting results of genetic studies. And we can take these fetal specific MQTLs and show that they're actually highly enriched for the genetic risk variants that are associated with schizophrenia. So the schizophrenia risk variants from the PGC at whatever significance threshold from GWAS that you take them are strongly enriched for these developmental MQTL sites. And just to show this is specific, you can take GWAS site findings from similarly powered studies of a neurodegenerative disease and also two diseases that are maybe not so implicated in the brain and show no significant enrichment amongst those sites. This tells us that the schizophrenia GWAS findings are really enriched for mediating gene regulation during brain development. And again, we've done very similar things using our RNA-seq data. So this is the paper just published in Gene and Biology with Nick Bray, again showing that just as MQTLs are highly enriched in fetal brain for GWAS variants, the same is true for expression QTLs as well. And we can also take these data and start using them to co-localize genetic effects on a trait and patterns of gene regulation. And this goes back to that question of fine mapping these big LD blocks that maybe harbour many genes and working out which is the actual causal gene in the context of disease. So here's a region of the genome from the PGC GWAS of schizophrenia, many genes in there, and our co-localization analyses highlight variation in this one specific gene, AS3MT, that we can then use to go on and do our functional characterization using cell models, and Emma's going to tell you something about that in the next talk. And there are many ways that we can put genetic and epigenetic information together. I'm not going to go into this in any detail. These are just two papers we recently published in the American Journal of Human Genetics using another approach called summary data-based Mendelian randomization, again with the aim of identifying overlapping genetic signals that are associated with both phenotype and regulatory variation. So putting this all together, I think it's really clear that it's not just genes and environment that play an important role in any type of complex disease, not just CNS diseases. I think this kind of third layer of gene regulation is going to be really important to carry on studying, but that we mustn't think of this as a kind of truly third entity, because in fact gene regulation is driven both by things in the environment but also by things in our actual DNA sequence, and actually provides a really nice mechanism, I think, for how those two things will interact and work together in the context of development to bring about disease. And in my final two slides, I just want to say why this is important, because I always think it's really good to contextualize, why we're doing this work and where it may be leading us in the context of making lives better for people with these really terrible conditions. And I think, one of the kind of real interests in studying gene regulation for me is the potential it gives us, unlike DNA sequence variation, for actually modifying the patterns that we see. And there's real interest now from pharmaceutical companies and biotech in targeting the different aspects of the actual epigenetic machinery, the molecules and the systems that actually act to lay down the epigenetic patterns, to actually read those epigenetic patterns, and ultimately actually kind of erase those epigenetic patterns. Because these are obviously really good targets for novel therapeutic strategies. And we're doing lots of work, longitudinal studies following individuals who are being given drugs that may be used to treat disorders like schizophrenia. This is work on a longitudinal study of DNA methylation changes in individuals, treatment-resistant schizophrenia patients, prescribed clozapine, just showing that just as smoking induces really big changes, so do these drugs. And actually understanding the mechanisms by which these drugs act is going to be really important for then developing new, better drugs that maybe don't have the negative side effects that these drugs do. And so in collaboration with Eli Lilly, for instance, we're doing a lot of connectivity mapping, taking different cell lines, dosing them with a whole range of neuropsychiatric drugs, primarily antipsychotics, and looking at the gene networks and patterns that are disrupted following exposure to the drugs, with the goal of firstly understanding how those drugs are working, because in many cases antipsychotic drugs were often discovered by accident, and their kind of mechanism of action isn't known. But also, because they have such severe side effects, designing drugs that do very similar things but maybe don't have the unwanted consequences of exposure. And Joanna's going to talk to you later today about some of her work on this, using zebrafish, again modelling effects of clozapine on gene regulation. Moving forwards, I think there's going to be many applications of epigenetics to personalised medicines. Personalised medicines are kind of real buzz thing now, but I hope I've convinced you that epigenetic signatures can give us really good biomarkers of disease. I think our schizophrenia blood work shows that. It can give us really good markers of exposure. I think our smoking, our clozapine work shows that. It can tell us about the mechanisms of disease. So, looking at this developmentally and brain development, or putting together genetics and epigenetic variation, telling us about how those mechanisms are operating. It can allow us to refine traditional risk models. Moving forward, it could well be provide us with biomarkers of response to drugs. And ultimately, as I said, hopefully we'll be at the stage where we can actually modulate the epigenome via certain interventions, be they pharmaceutical or more lifestyle. So again, just want to finish with a slide of my group. These are the people who do all this work. A really fantastic group. We're always looking out for students and postdocs to come and work with us. Our door's always open if people want to come and spend a bit of time with us and learn about what we're doing or train on certain things. We'd be really delighted to host people in our lab in Exeter. And if anyone has any questions, please just come and ask me later or in coffee or whenever. But thank you very much.